Hello guys, you're welcome to this session where we want to discuss about hypersensitivity reactions. Sometimes we interchangeably call them allergic reactions, and this is immune. It is body's immune to react against foreign antigens. So hypersensitivity comes from two words, hyper and sensitivity, whereby hyper means exaggerated. So hyper means something which is exaggerated. Then sensitivity is responding to the stimuli, something you are not used to. So we are seeing hypersensitivity reactions. These are exaggerated immune reactions. Exaggerated immune reactions which are triggered in response to foreign antigens. So they are triggered in response to allergens. Sometimes we call them allergens, or what we call foreign antigens. So something must be triggering this exaggerated reaction. That's why we call them hypersensitivity. So a hypersensitivity, by definition, these are exaggerated immune reactions which are triggered in response to allergens, or what we call foreign antigens. And these hypersensitivity reactions, they are divided into four. So there are four types of hypersensitivity reactions which we want to discuss in this session. And as we begin them, we always remind you to subscribe. Always remind you to like the video when you have learned, and share the comments that we may continue to learn more and more. So hypersensitivity reactions, or what we are calling allergic reactions or exaggerated immune reactions, they are categorized into four types. And these types include type 1, type 2, then type 3, and type 4. Whereby type 1 to type 3, they are antibody mediated. These ones are humoral, or what we call antibody mediated. Whereas type 4 is cell mediated. So type 1 to type 3, they involve antigen-antibody reactions, whereas type 4 is the one which is cell-mediated. That one is very important to note, that type 1 to type 3, they are antibody-antigen-mediated, whereas type 4 is cell-mediated, whereby we shall see it will be mediated by T helper and T cytotoxic. Whereas these ones are antigen, and this antigen majorly we see involved is IgE for type 1, then for type 2 and 3. So type 1, it will be having IgE, whereas type 2 to type 3, type 2 and type 3, they involve IgG and IgM. So as we begin with the type hypersensitivity reaction type 1, for it, sometimes you can call it an anaphylactic reaction, sometimes we call it anaphylactic reaction, or if you don't call it anaphylactic, we can call it immediate hypersensitivity reaction. So you can call it immediate, or we can call it anaphylactic reaction. And this type 1, or what we call type 1 hypersensitivity reaction, is mediated by an antibody. This antibody we have seen is IgE, and this IgE binds either to mast cell that is in tissue, or binds to the basophils in blood. So the cells which are involved is the mast cells in the tissue and the basophils in the blood, whereby IgE binds to them using the F portion. So after seeing that, that when I have a mast cell or a basophil, this is a mast cell or a basophil, so this basophil has the Fc receptors, and after having the Fc receptors, the IgE comes and binds. So we start to see the IgE binding, and another IgE binds using the Fc portion, and this is the Fc receptor. And this one is triggered by an allergens or anything that is foreign. For example, it can be dust, it can be pollen, it can be food you have eaten which the body cannot tolerate. So what happens is that this IgE, in the presence of an allergen, they bind to the Fc receptor of the mast cell, and after binding to the Fc receptor, they come and conjugate the antigen, the foreign antigen that is triggering. It can be an antigen, or you can call it an allergen. It is complexed in between the two IgE. So these are the IgE. This is IgE, this is IgE. Binding to Fc receptor using the Fc portion of the antibody. And after them binding, they neutralize the antibody or the antigen, and in so doing, they trigger the mast cell or the basophil they cause the degranulation, and this degranulation of the mast cell or the basophil results in two release of inflammatory mediators. So it releases inflammatory mediators, and these inflammatory mediators include, we have leukotrienes, we have thromboxane A2, we shall have histamine being released, we shall see platelet activating factors being released, we shall see serotonin being released. So these are some of the inflammatory mediators that are released when the allergen binds to the two IgE antibodies using the Fc portion on the mast cell or the basophils. And after binding, it causes degranulation leading to release of inflammatory mediators. And these inflammatory mediators, we have seen leukotrienes, and the major importance of leukotrienes is chemotaxis. They play a big role in chemotaxis. That is the election of flow of the cells or the phagocytes towards the allergen. Then thromboxane is the vasoconstrictor. Thromboxane and serotonin. We shall see if I can shift this thromboxane here. Thromboxane A2 and serotonin, these ones cause vasoconstriction of blood vessels. Then histamine is the one that triggers inflammation also. So these guys participate in inflammation. They participate in inflammation whereby platelet activating factor, they also activate the platelets at the site where the allergen is, and then histamine also playing a big role in initiating these allergic reactions. So these are the inflammatory mediators that are produced to enhance inflammation or allergic reactions at the site where the allergen is. And after seeing these inflammatory mediators being released, here the end result of the anaphylactic reaction. Examples of anaphylactic reaction include urticaria, that is rash, urticaria or getting some skin rash after an allergen, or you have eaten some meat which the body responds to, you have taken maybe some drink which your body is not compatible with. We see urticaria coming in, or what we call body rash on the skin. Sometimes we normally see asthma. Whenever you are not used of coldness or you are exposed to some allergens, you inhale them, we see asthma attacks coming in. And many other allergic reactions are part of type 1 hypersensitivity reactions. So the major The major examples here, we are seeing urticaria, that is skin rash. We are seeing asthmatic attacks being examples of hypersensitivity type 1, which we have called immediate hypersensitivity reaction, or we can call it anaphylactic reaction. So many times when this reaction is, sometimes you see even a cough coming in as a defense mechanism towards this. So this is what we can discuss about type 1. So what we need to remember about type 1 is that the antibody which is involved is the IgE, which binds to the Fc receptor on the mast cells or the basophils, complexing or neutralizing the allergen, leading to degranulation, which triggers production of inflammatory mediators, which we have listed here. And then examples include urticaria, that is skin rash, asthma attacks are the examples of hypersensitivity type 1 reaction. So after seeing that, and sometimes we have also added other allergic reactions being part of the hypersensitivity type 1. So the next type we can talk about is type 2. And type 2, as we have seen in type 1, it is IgE. In type 2, it involves antigen-antibody reaction, antigen-antibody complex. And this antigen-antibody complex is triggered by the presence of either IgG or IgM. That in the presence of IgG or IgM, these guys are able to activate the complement system. They activate the complement system. And this complement system, as we remember, or if you haven't watched my video on the complement system, you can visit it. We shall see that this complement system, whenever it is activated, it has some intermediates like complement 3B, it has complement 3A and 5A, and it also finally it forms what we call the MAC. So these components of the fixed complement system by the IgG or IgM, and before we go far, that this type of reaction is also known as cytotoxic hypersensitivity reaction. Cytotoxic hypersensitivity reaction, and this reaction being cyto means cell, so it is cell specific. So if they ask you in an exam that which of the following type of hypersensitivity reaction is cell specific, that is type 2 hypersensitivity, which is triggered by IgG or IgM, that binds and fixes the complement system, leading to production of complement 3B. And complement 3B is a strong complement 3B, causes opsonization, that is tagging the infected cell, whereas complement 5A and complement 3A, these guys enhance phagocytosis by causing chemotaxis, which at the end enhances inflammation. Then this opsonization enhances the phagocytosis of these allergens, whereas the MAC, which is produced after the complement has been fixed, is responsible for cell lysis. So we are going to see that whenever the complex is formed and the complement system is fixed, it produces the MAC, and the MAC comes and binds on the cell surface, and when the MAC comes and binds on the cell surface, it starts creating pores in the cell wall of the cell, and when it creates pores, it leads to the leakage of intracellular content. And when intracellular content leaks via the pores created by the MAC, it leads to what we call cell lysis. So this is this is type 2 hypersensitivity reaction, whereby the major thing we see it involves IgG, IgM. fixing the complement system, leading to production of complement 3B, which is an opsonization, which does a big role in opsonization, whereby it enhances the phagocytosis. We see production of complement 5 and complement 3, which are responsible for inflammation by enhancing chemotaxis, and the MAC that goes on the cell membrane and causes cell lysis by creating pores. And whenever MAC creates pores, and remember MAC, what we call membrane attack complex, comprises of complement 5, 6, 7, 8, and 9. This is what forms the membrane attack complex, which goes on the surface of the cell membrane and creates pores, leading to leakage of intracellular content, hence cell lysis, or that is what we call cytolysis. So cell lysis, we can call it cytolysis. And that's why we say it is cytotoxic hypersensitivity reaction specific to the cells. So that is what we can discuss about type 2. And examples of hypersensitivity type 2 include hemolytic disease of the newborn. It includes hemolytic anemias, and these hemolytic anemias can be, they are part of type 2 reaction. We find another example as blood transfusion reactions. Blood transfusion reactions. Whenever you are given a wrong blood group or a resource which is not compatible, we see cells in the blood being reactive. That is blood transfusion reactions. And sometimes we see an example as Graves' disease being an example. And sometimes we can see myasthenia gravis. Myasthenia gravis. So myasthenia gravis is also an example of type 2 hypersensitivity reaction, whereby hemolytic disease of the newborn is due to rhesus incompatibility, rhesus factor incompatibility of the mother and the fetus, whereas hemolytic anemias, this cause increased cell lysis, that is red cells being destroyed. Then blood transfusion reactions, when you are given a wrong blood group, and Graves' disease, this is whereby the body is producing autoantibodies, which resemble thyroid stimulating hormone. So these autoantibodies come and destroy what we call the follicular cell of the thyroid gland, leading to hyperthyroidism. That is what we call Graves' disease. Then myasthenia gravis, this is whereby the body is producing autoantibodies, the antibodies which are against self cells. And these antibodies, they go and attack the sarcolemma, or what we call the neuromuscular junction. We know at the neuromuscular junction, that's where we see impulses from neurons going into what we call the muscles. That if we have a neuron synapsing with the muscle, which is a skeletal muscle containing the actin and the myosin, this is the sarcolemma, and the receptors of the nicotinic receptors are found on the sarcolemma. So we find the body is producing autoantibodies that come and destroy the nicotinic muscular receptors. And when they destroy the nicotinic muscular receptors, people have muscular weakness and paralysis. That is what we call myasthenia gravis, which is a type 2 hypersensitivity reaction. So this is what we can discuss. So always tell us what triggers it. You tell us how it is brought about and give us at least 3 examples. And in objectives, we can always ask you that the following reactions are type 2, except. So you should be able to tell us different examples of reactions under each type, so that we can be able to pass our exams. Then type 3 hypersensitivity reaction, this is immune complex. This is, sometimes we call it immune complex And this type of reaction, it is tissue specific. It is tissue specific. And after being tissue specific, involving IgG and IgM, it involves IgG and IgM, meaning it is antibody mediated, whereby these guys bind to the antigen or the allergen, causing what we call, so they involve what we call soluble antigens. They involve what we call soluble, they react to soluble antigens, whereby they react to form antigen-antibody reaction or complex. Antigen-antibody immune complex, and this antigen-antibody immune complex formed by the soluble antigens, which are circulating within the body. They might be localized at the site of the antigen, or they might be moving in saturation. So what happens is that this immune complex, when it is deposited on the blood vessels, on the endothelial cells of the blood vessels, it comes and fixes the complement. It also fixes the complement system. And after fixing the complement system, which we have seen, production of C3b, C3a, and MAC in the blood vessels, in the endothelium of the blood vessels, or at the site where they are localized. So these reactions or immune complex, sometimes they are localized in one site at the tissue site, or they are moving in saturation, and they are deposited in the endothelium of blood vessels. And after being deposited, remember they fix the complement, leading to production of MAC. And when MAC is produced at the endothelial cells of these vessels, it creates the pores, it causes perforation of the blood vessel, leading to leakage of intracellular content, leading to tissue damage. So the end result of this reaction leads to tissue damage by the complement system, whereby the soluble immune complexes, they are either localized at the site or they are being moved at the saturation, and they end up being deposited in the endothelial cells, which causes pores, leading to tissue damage. So the end result of type 3 reaction is tissue damage by the soluble antigen-antibody complement complexes, which fix the complement system, leading to production of membrane attack complex, which causes perforation of the vessels, leading to tissue damage. And if they are localized at the tissue level, they also, the MAC goes and causes tissue damage. And examples of these reactions include it includes systemic lupus erythematosus is an example. It contains rheumatoid factor. Rheumatoid factor is also what we call rheumatoid arthritis. This rheumatoid arthritis affects the joints, leading to inflammation of the joints due to the deposition of the soluble immune complexes. And another example we can see is good pasture syndrome. Good pasture syndrome is also an example of type 3 hypersensitivity reaction. Another one is Arthur's reaction. Arthur's reaction is also an example of type 3 hypersensitivity reaction. So Arthur's reaction, we have good pasture syndrome as an example. We have rheumatoid arthritis and systemic lupus erythematosus. These are the examples, 4 examples you can give under type 3 hypersensitivity reaction. So whenever you're asked in an exam that outline NF4 examples of type 3 hypersensitivity reaction, do not fail to list these ones. Or if they are brought them in objectives, we should be able to pass them. They can say the following are type 3 hypersensitivity reactions, except we put 3 of these and we mix in the ones of type 2. So you should be able to pass that one. And finally, the last And we have seen the first three are antibody mediated. And the last, which is type 4, is cell mediated, as we said from the beginning. So being cell mediated, meaning this one is cell mediated, or sometimes we call it delayed hypersensitivity reaction, meaning for it takes long to act. That's why we call it delayed. And it is the cell mediated. For it is the only one which is cell mediated, whereby it involves T helper cells and T cytotoxic cells. These are the part of the T lymphocytes. Sometimes these ones we call them CD4 positive cells. These are CD8 positive cells. So what happens is that in the presence of an allergen, or in the presence of an antigen that is triggering this reaction, they trigger the production or proliferation of the T lymphocyte to produce the T helper and the T cytotoxic by the lymphoid tissue. And when they are produced, we see the CD4s, they do a great role of producing cytokines that whenever I have an antigen presenting cell, and this antigen presenting cell is infected with maybe a virus or a bacteria from maybe you have a bacteria from extracellular fluid which is engulfed, and after being engulfed, it is broken down into smaller peptides which are presented by the major histocompatibility complex type 2. If it is a bacteria from extracellular, it is presented by major histocompatibility complex type 2, which presents the peptide or a portion of the peptide from the bacteria on the surface, which comes and the CD4 come and bind. So this is the CD4 positive cell which comes and bind, or causing what we call opsonization. But it does not only cause opsonization, it also produces what we call cytokines. And these cytokines, like tumor necrotic factor and interleukins, are responsible for activating macrophages. So these cytokines activate macrophages, and also they activate the cytotoxic. So they come and activate the cytotoxic, what we are calling the CD8 positive cell. And after activating them, these two guys come and kill the infected cell. They come and kill the infected cell. And sometimes these guys also trigger inflammation at the site. They also trigger inflammation. So the end results of type 4 hypersensitivity reaction involving CD4 and CD8 is cell killing of the infected cell with the antigen. The antigen might be the extracellular bacteria, or it might be the intracellular virus. And if it is intracellular, the antigen presenting cell, if it is a virus, it breaks its portions and presents them on a major histocompatibility complex type 1, presenting them to the CD4 cell, which produces cytokines, calling the cytotoxic killer cells or the T killer cells and the macrophages to come and destroy the virus infected cell. So the major role of CD4 is opsonization and the production of cytokines or chemokines that cause or that activate the CD8 and the macrophages, which kill the cell, and at the end run, they also trigger inflammation at the tissue. And examples of type 4 hypersensitivity or delayed hypersensitivity reaction, we have what we call tuberculin test. Tuberculin skin test is done to people with tuberculosis, Mycobacteria tuberculosis. That whenever someone has Mycobacteria tuberculosis, and we are suspecting them to have, we can do tuberculin test, which is done on the skin by making some prick, then we add an antigen, which we then, at the end, we monitor the size of the inflammation. And when the size of the inflammation is big, we can confirm that someone is Mycobacterium tuberculosis positive. And another example is contact dermatitis. Contact dermatitis. And lastly, we can see even allograft rejection. Allograft rejection, whenever you give a tissue transplant, sometimes this cell-mediated hypersensitivity reaction is the one that causes allograft rejection. So these are the 3 examples of type 4 or cell-mediated or delayed hypersensitivity reactions. So this is what we can discuss today about hypersensitivity reactions. Hope you have learned something, whereby we have discussed type 1, type 2, type 3, and type 4. We have seen type 1 is an anaphylactic immediate hypersensitivity reaction involving IgG and mast cells or the basophils, which produce inflammatory mediators leading to inflammation or to removal of the allergen. And examples we have given: rash or urticaria, asthmas, and allergic reactions. And type 2, we have seen it is cytotoxic, meaning it is cell-specific and involves IgG-IgM that fixes the complement system, and the end result is cytolysis, phagocytosis, and inflammation of the cells, because it is cell-specific. And we have seen examples like hemolytic disease of the newborn, hemolytic anemias. We have seen blood transfusion reaction, Graves' disease, and myasthenia gravis. And we explained how it comes about. In type 4, we have seen it is tissue-specific, and being tissue-specific, that it involves IgG-IgM, which is responding to soluble antigen, forming immune complex. And this immune complex, we have seen it can be localized or in saturation. And when it is in saturation, it is deposited in the endothelium of the blood vessels, leading to tissue damage by the complement, what we call membrane attack complex. Examples we have seen: systemic lupus erythematosus, rheumatoid arthritis, Goodpasture syndrome, and Arthur's reaction. Then the last one is delayed hypersensitivity reaction, or what we have called type 4, which is cell-mediated. Others are antibody-mediated, but for it is cell-mediated, involving CD4 and CD8, which we have seen all of them work together to bring about inflammation and the killing of the antigen. And we have seen examples being tuberculin test, contact dermatitis, and allograft rejection. Some books also add here farmer's lung disease, but sometimes it might be part of type 3. Thank you so much for listening from the beginning until the end. Always remember to subscribe at muhumuzamedicalsciences.com. Hope you have learned something. Thank you so much.